Hello, welcome to the Proteomics video series, featuring interviews with three leading experts who'll share insights into the many facets of the proteomics field. Today, I'll be chatting with Dr. Hassan Gharibi, who's the manager of the single-cell proteomics core facility at Karolinska Institute in Sweden, to learn more about how single-cell proteomics is enabled in a core facility setting, from supporting researchers and managing workflows to implementing cutting-edge mass spectrometry technologies and driving innovation in high-throughput proteomics. Hi, Hassan. Thank you so much for joining us today. So I was wondering if you could begin by briefly introducing yourself, your role at the single-cell proteomics core facility at Karolinska Institute, and the main scientific goals of the platform.
Hi, Laura. Thank you for having me. I'm Hassan Gharibi. I'm the manager of the single-cell proteomics core facility at Karolinska Institute in Sweden. And simply put, our main goal is to enable researchers across the world to look at the proteins at single-cell level, or if they have like samples that have low amount of protein, like inherently, like embryo samples. They're not actually single cells, but they give you little amount of proteins.
Lovely. And now we've seen that single-cell proteomics is rapidly evolving. What are the biggest technical and analytical challenges in measuring proteins at single-cell resolution, and how is your facility addressing them?
Yeah, sure. I would say like the first thing is that the amount of the protein is extremely low. We are working with around 200 picogram of proteins that we get from mammalian cells. And also like the instruments are being more sensitive these days with the advancement that we have from the past at least 2 years. But still the sensitivity is a problem. And won't be able to get the full proteome, like the whole proteome as in bulk samples, but still that's one thing. And the other thing is the volume of the samples, because we want to also work with extremely low volumes because you don't want to dilute the samples that much. But still at this point, even a small, I would say, error in pipetting, that could give you like 50% loss of your sample, which is something that you don't want to have. It's best to run with the most sensitive instrument. After calibration, like you have to have your QC steps in place. And for the volumes, I would say it's better to automate everything that is possible. Like at the moment, like we have the single cell dispenser that are automated, the digestion process can be automated, the labeling process if you want to do the multiplexing.
Amazing. So from a practical perspective, what happens when a researcher brings a project to your facility? Could you walk us through a typical workflow from experimental design and sample preparation to mass spectrometry analysis and then data processing?
Yeah, absolutely. So we use a platform called iLab for submitting the project description from the customers and clients. Also, they can reach out via email, but generally we have that form in place. And in the project description, they need to include what is their objective and goals for the experiment, if they have prior data for the samples that they want to send us, the samples that they want to analyze, and also if they have any sort of treatments that they want to apply to it. If it's proteins, we are booking a consultation meeting with them, and we will discuss their actually objectives, because sometimes you need to kind of reformulate the questions, like the biological question that they have, to what proteomics can answer. Sometimes you need to come up with another, like not a different, I would say, question, but a different angle that would answer the question. And sometimes we have the methods like already in place for their needs, but sometimes we might not have it. In such cases, we offer a pilot study, which is a free of charge, I would say, part of the work, and we do the feasibility study first. If it yields the good result and the criteria that we need to meet, then we go and do the full scale experimental design with the clients. And at this point, generally they ask for like different instrument, which are the things that we kind of like advise them what kind of instrument you need to use, what would be the pros and cons. And like generally from the client point of view, the most important thing is to look at the depth of the proteins or the throughput of the samples. So we kind of like balance between this for them. And then we send the code. And if they agree, then we get the samples from them. Generally, they can send us the samples. But yeah, so at that point, we get the samples, we do the sample prep from the standardized protocols that we have for a given method. We do the QC. We use everything new for any new clients. And at the end, we do the data analysis, not only just giving them the protein abundances, but based on their question, we would write a whole like a HTML interactive report that you can browse the QC steps that we had in place, the actual findings, and since we also like do like pathway analysis based on like the proteins that we saw, you can kind of get a better, I would say, insight of not just like looking at the protein, but what does it mean biologically. So yeah, that would be that.
Wow, that sounds like an amazing workflow. So your work also involves developing standardized data processing pipelines and cross-facility comparisons. Why is reproducibility such a challenge in proteomics, and what strategies help ensure consistent results across experiments and laboratories?
I would say so the reproducibility and the consistency. I would say like it's like comparing like the RNA sequence and the DNA sequence is like mostly based on Illumina and nanopore. So it's like comparing different, I would say, different cars from like the same kind of like a car from different manufacturer. Like if you compare like an Aston Martin to a Bentley, you see a bit small difference, but there is. But in proteomics, the difference in the instrument, like the LC systems, the method, the pipeline that you use, the acquisition method that you use, and even like the sample preparation that you use, the way that you search the data eventually, and so on, it's like comparing like planes and boats and car. Like they are totally different. So it's kind of hard to compare different laboratories if they are using different instrument, if you use different gradient, different method, and so on. So like it's gonna be hard because you actually are measuring different things. Like if you use time of flight is different than if you're looking at the orbit trap. It's like a totally different, I would say, like analyzer. Not that you're measuring different thing, but you're measuring the same thing in a different way. For this, at least in our lab, what we do is that we have a certain like software that we always use for like searching. The parameters always set. We don't change those parameters between different experiments unless it's needed. The instruments is always calibrated. The same instrument is being used for the same experiments. So that's how you need to make it. Otherwise, there are so many parameters that you can play with it in the proteomics that would totally give you a different result.
Yeah, lovely. Thank you. I love that analogy as well. So now many researchers entering proteomics come from biology rather than analytical chemistry or mass spectrometry. How do you guide collaborators, especially early career researchers, when designing experiments that are realistic and technically robust?
Yeah, actually, that's a very important question. I was part of a chemical proteomics core facility before I became the manager of the others core facility that we have. And yeah, in the meetings, we can actually see like that people, they have a good question, but it's not about like, you don't know if you can actually measure it with mass spec. So they don't have the expertise in the mass spectrometry. The same for me, like 8 years ago, I didn't know anything about mass spec. So, but what we need to make, at least for them, is to kind of make a realistic question that can be answered by mass spec or proteomics, and also to do like an expectation management here, because like you're not supposed to see like 20,000 proteins. No one can actually see that. It's not like doing the genome sequencing. So what we do is that to make sure the question can be answered, first of all, with proteomics, and then we help them to do a proper experimental design, like what kind of control you should use, if multiplexing would be better or compared to label-free analysis. Like these are like changing that based on like how many samples you want to analyze. That would be a bit different. And yes, and also like throughout the whole I would say process, we also help them with the data analysis as well. Like we always help them with the interpretation. We do the statistical analysis, biological analysis, and the pathway analysis. So it's just not like that they're gonna end up with a bunch of proteins. So at some point we would like get involved heavily to make sure that they are answering the right question that we can give a good output for.
Lovely, thank you. And I'd love now to talk about the process once data is acquired, as proteomics data sets can be pretty complex. How does your team help researchers interpret the data and translate mass spectrometry outputs into meaningful biological insights?
Yeah, as a former bioinformatician, I can say that the proteomics data could be scary if you're working with like large cohorts, and if you have like so many missing values and so on. So I would say the way that we, as I said, help the researchers is that when we report the final findings, we give an interactive HTML report. It's not like an Excel file that you need to go through and so on. We have everything that you can click around, you can export, and you can look at the data from your own perspective. But the structure of the report is that you have the QC steps, so that we make sure that we get a meaningful result. Like if something failed, it's either could be like experimental design that we failed on it. Maybe we didn't do a good, let's say, sample prep and so on. So we make sure at this step that everything that we see is up to the standard that we wanted to have. And after that, we do the statistical test for them. We look at the protein, I would say, relevance to the biological questions that they have. Not only just giving names, we look at the pathways, we look at the clusters that we get. And throughout the whole work, we try to make sure that what they were like looking for, like what they're after for, can be answered with this data.
Perfect. And now, looking ahead, how do you see single-cell proteomics shaping fields such as systems biology, drug discovery, or precision medicine? And I wonder if you have any words of advice that you'd give researchers who want to leverage these technologies effectively.
I'd say, of course, like the system biology could be affected by the single cell proteomics because it's like looking at the omics data. So now we have transcriptomics, genomics. This is another level and layer that we are adding. It's like a functionality of the cells, which of course is going to impact the field. As for the drugs, I would say the significant thing that the single cell proteomics can bring is the fact that you're actually looking at the functions that is happening in single cells, and you can look why cells from different states are behaving differently to the same drug. So you kind of can see if some cells are resistant, some are sensitive, to what degree they are sensitive or resistant, and what could cause this. So you can actually make a better drugs or make a better dosage. Now, with precision medicine, you apply something to a specific person, but still we do it as a bulk person, like whatever that we have inside that person, it would be like bulk and averaged. So if we do single cell of an individual, like in precision medicine, is like looking at the precision medicine, like it's like precision plus medicine, that you can make a great profile, like a full profile of all the cells of an individual, and then give a treatment. So I would say like that could be quite like revolutionary for that sense. As for the I would say advice to anyone, okay, I'm quite like young to give any advices, but I'd say that like yeah, it's gonna be a huge thing, single cell, spatial proteomics, single cell, these things that are being kind of spearheading these days. Like I think like 4 years ago we were quantifying up to like maybe 1,000 proteins. Now we have like even 5,000 proteins with different methods and so on. So you need to properly make an experiment design. Otherwise, like if you change something, you won't be able to understand what does the results tells you. So you need to have a proper experimental design before going and using these tools. So I would say like making a good question that could be answered by the single cell proteomics would be the first thing to look at.
Lovely. Hassan, thank you so much for sharing your experiences and your insights with us today. It was a pleasure speaking with you.
Thank you for having me.
Thank you for watching this episode of the Proteomics video series. If you haven't already, be sure to check out the other 2 videos in the series, where we explore additional perspectives and topics from leaders in the proteomics field, including proteomics and diagnostics and building reproducible pipelines for proteomics data. See you next time.